So, hello aspirants. In today's video, we will discuss the rest of the portion of cell cycle regulation. And if you haven't watched my previous video, the part 1 of cell cycle regulation, you must go through that and then return to this video in order to get the proper concepts out of it. Now, our last slide was this, where we have discussed regarding the regulation of cyclin B degradation. Now, cyclin B degradation, as we all have discussed previously, is based upon a N-terminal destruction box. And that N-terminal destruction box, it undergoes ubiquitination, or it is rather the site of ubiquitination, where ubiquitin units are bound by the activity of APC by C in association with CDH1. Here, in this slide, we have mentioned how CDH1 is regulated and how come it is activated by means of a phosphatase activity CDC14. So, coming to the next part of the discussion. Now, let's see, we have our cyclin and CDK. The cyclin and CDK, they togetherly form the MPF, the mitosis promoting factor. We all are aware of it. And based upon differences in certain types of organism, whether it be Saccharomyces cerevisiae, whether it be mammalian system, or whether it be Saccharomyces pombe, the MPFs, that means the cyclin and the CDKs, they vary from each other. Now, what actually happens when it comes to cyclin and CDK, when a cell is supposed to be at G0? From G0, the cell enters into G1. As simple as that. So, for a cell to enter into G1, the first thing when it comes to mammalian cell, the first thing that happens is activation of cyclin D and CDK 4 by 6. This is the typical case. Now, how come this kind of activation takes place? Or rather, how CDK is getting activated in order to perform the catalytic activities thereafter. So, in order to determine that, let's see over here. This is our mitotic cyclin. This is our CDK, as all you can see. Cyclin, for Saccharomyces pombe, it is CDC13. For Saccharomyces cerevisiae, it is CLN3. And for mammalian, cyclin D. Same goes for CDK. For pombe, it is CDC2. For cerevisiae, it is CDC28. And for mammalian system, it is CDK 4 by 6. Now, the cyclin and CDK, they are associating with each other. Before the entry, what happens is there lies a regulatory protein. There lies a regulatory protein called V1. What V1 does? V1 phosphorylates. What V1 does? V1 phosphorylates a specific residue. V1 phosphorylates a specific residue at CDK. And that phosphorylation occurs at tyrosine 15. This kind of phosphorylation at tyrosine 15, this kind of phosphorylation at tyrosine 15 by CDK1, V1, This is kind of an inhibitory approach. Means when cyclin and CDK is present, V1 comes into action. What V1 does, it phosphorylates. This is our cyclin, this is our CDK. V1, what it does, it phosphorylates CDK at tyrosine 15. It phosphorylates at tyrosine 15. This type of phosphorylation, this type of phosphorylation is inhibitory. This type of phosphorylation is inhibitory. That means by the activity of V1, now the cyclin and CDK are at static state. They cannot regulate the cell, enter into G1 or to enter from G1 to S or from G2 to M. So what can be done in order to overcome this kind of an issue? Then comes the function of CAK. Then comes the function of our CAK, cyclin activating kinase. What cyclin activating kinase does, it again phosphorylates. Cyclin activating kinase CAK, it again phosphorylates threonine at position 161. It activates, rather phosphorylates threonine at 161. So initially there lies a phosphorylation at tyrosine 15. This kind of phosphorylation is inhibitory and it is performed by V1. Next comes the activity of CAK. CAK protein, what it does? It activates or rather phosphorylate threonine within CDK at position 161. Now when this kind of phosphorylation occurs, that will lead to the introduction of another kind of protein, which is a phosphatase. So comes the function of a phosphatase, which is CDC25. So let's see. Here we have our cyclin and CDK that is phosphorylated at tyrosine 15, which is an inhibitory approach. From this, another regulatory protein, CAK, comes into action. What CAK does, we have our cyclin, we have our CDK, as it is phosphorylated at tyrosine 15, which is an inhibitory approach. Now by the activity of CAK, phosphorylation at threonine 161, phosphorylation at threonine 161 takes place. Now this kind of phosphorylation is rather promoting the entire thing, is rather promoting the cell cycle. But there lies an immense problem. The problem is tyrosine 15, as we all know, is an inhibitory protein, is an inhibitory approach. So phosphorylation at tyrosine needs to be removed. Phosphorylation at tyrosine 15 needs to be removed. Threonine 161 phosphorylation is activating the CDK cyclin complex, but phosphorylation at tyrosine 15 is inhibiting it. So we have to remove it. For the removal, there comes the activity of a phosphatase. There comes the activity of a phosphatase, which is CDC25. CDC25, what it does, we have our cyclin, we have our CDK. So, being a phosphatase, CDC25 being a phosphatase, it dephosphorylates, it dephosphorylates the tyrosine 15. Tyrosine 15 gets dephosphorylated. But our threonine 161 is still phosphorylated. Threonine 161 is still phosphorylated. So, Cyclin-CDK V1 comes into action. It phosphorylates the tyrosine 15, which is an inhibitory approach. Then comes CAK, which phosphorylates CDK at threonine 161, which is a approach which is kind of activating the entire mechanism. But if tyrosine 15 remain phosphorylated in this manner, then the entire cell cycle won't be able to proceed. So therefore comes the activity of a phosphatase called CDC25. What CDC25 does, it dephosphorylates tyrosine 15. As a result, the entire cell cycle can proceed by means of activation of cyclin and CDK. Now, this is a very simple approach. There lies two questions. One, how come only the tyrosine 15 gets dephosphorylated? Threonine 161 is also phosphorylated. So CDC25 can also act upon threonine 161, but that doesn't really happens. What is the logic behind it? This is my first question. And second question is, apart from tyrosine 15, there lies another regulatory unit. There lies another amino acid, threonine 14, threonine 14, which acts as an inhibitory factor within CDK. So, comment below the answers regarding these two questions. So, my first question is, I'm repeating myself. Question number one, how come only tyrosine 15 gets dephosphorylated? Why not threonine 161 is also getting dephosphorylated by means of CDC25 phosphatase activity? This is my question one. And my question number two is, apart from tyrosine 15, there lies another inhibitory approach by means of threonine 14. State these two answers in the comment section. And if some of you are unable to answer it, let me know that also. I will definitely prepare a separate thought regarding this aspect. So this is kind of the activating factors that are associated with Cyclin-CDK regulation. Now the fact is, we have our V1. Let's see. We have our V1. We have our CDC25. Both of these proteins are regulatory. V1 it initiates phosphorylation, and CDC25, it promotes phosphatase activity. Now let's see. Suppose we are considering a Saccharomyces pombe cell. If that particular cell, if that particular Saccharomyces pombe cell shows mutation at V1, I'm repeating myself. Suppose we are considering a Saccharomyces pombe cell, and within that cell there lies a mutation at V1. Then what will happen? Now V1, the function of V1 is initiating, the V1 function is like restricting the cyclin and CDK activity. V1, by phosphorylating at tyrosine 15, it restricts the activity of cyclin and CDK. So if a if a particular cell 1 is pombe, it shows V1 mutant. It shows V1 mutant. Then what can be the possible approach, or what can be the possible outcome for that cell? Now let's see. This is our typical cell. Suppose within that cell, cyclin and CDKs, they are now, because of V1 mutant, cyclin and CDK, they are cyclin and CDK specified for Saccharomyces pombe. They are not getting hindered, no inhibition by V1, as V1 is mutated. So cyclin and CDK, there is no V1. As a result, CAK will act upon it. As CAK will act upon it, CDK will be phosphorylated at threonine 161 very easily. And as there is no inhibition, by V1 at tyrosine 15, CAK, by phosphorylating threonine 161, will readily promote the cell for division, As a result, if this is our cell, then this particular cell, before attaining its proper size for division, this particular cell, before attaining its proper size for division, it will undergo division, forming two small cells. So this particular cell of Saccharomyces pombe, where V1 is absent or mutated, that means V1 is practically restricting. So what is the function of V1? V1 is practically restricting a cell to attain its proper size so that it can enter the divisional phase. So if V1 is absent, then the cell will readily enter into divisional phase. And hence, before attaining the proper size, it will undergo division. Just like this. So, this is our wild type. So, within wild type, wild type means both V1 and CDC25 both are present. V1 and CDC25 both are present. There is no problem with it. So, it will undergo division forming two distinct cells. The problem lies with V1 mutant. As you can see, V1 mutant will generate cells which are smaller in size compared to wild type. Why? Because V1 rather hinders the cell so that the cell can prepare itself or enlarge in its proper size and hence can undergo division. So if V1 is absent, then CAK will function readily and it will phosphorylate threonine 161, and as V1 is not present, hence tyrosine 15 will not be phosphorylated. So CDC25 activity won't be required for this kind of a mutant and hence the cell will readily divide. I'm repeating myself, V1 mutant. So for V1 mutant, tyrosine 15 is unphosphorylated. CAK will phosphorylate at threonine 161, which is an activating function. Now, as tyrosine 15 is unphosphorylated, the CDC25, which is a kind of phosphatase that removes the phosphate group from the tyrosine 15, that won't be able to perform its activity, or rather it is not required in this case. As a result, the cell will readily undergo division forming smaller sized cells. On contrary to that, if the third condition CDC25 is mutated, then what will happen? If CDC25 is mutated, then let's go back to my previous slide. Now, suppose this is our cell. Within cell of Saccharomyces bombay. There lies our cyclin as usual. There lies our CDK. So V1 is present over here. Here the mutation lies at CDC25. CDC25 mutant. So V1 is present. So if V1 is present, then definitely there will be phosphorylation at tyrosine 15, which is an inhibitory approach. There will be phosphorylation at tyrosine 15, which is an inhibitory approach. Now, by this time, what will happen? CAK will come into action. CAK, as we all know, apart from tyrosine 15 phosphorylation, CAK, what it will do? It will phosphorylate our threonine 161. But the cell will be restricted at this stage. Why? Because we don't have our CDC25. If CDC25 is practically absent within the cell, then phosphatase activity won't be able to be performed upon tyrosine 15. Phosphatase activity won't be able to be performed upon tyrosine 15, and hence the cell will remain as it is without division. The cell will remain as it is without division because CDC25 is absent, and without phosphatase activity, the cell cannot enter the M phase. Without phosphatase activity, the cell cannot enter the M phase. So the cell will enlarge. The cell will enlarge, but it won't undergo division. The cell will enlarge, but it won't undergo division because CDC25 cannot perform the phosphatase activity upon tyrosine 15. So both tyrosine 15 and threonine 161 will remain phosphorylated in this condition. So CDC25, the cell will enlarge. So this two things are very important when it comes to CSIR net. Just try to pay attention while solving the problems. Now, moving to my another concept. Previously, in my previous class, I have mentioned that the chromatids, during cell division, we all know that chromatids, they separate from each other. That is the purpose. The chromatids must separate from each other so that anaphasic movement can take place and the chromatids can be aligned on two distinct poles. Now, suppose this is our chromosome within cell. This chromosome, the two chromatids, chromatid 1 and 2, they need to be separated from each other. That is the purpose. But before anaphase, they aren't able to differentiate from each other. What is the reason behind it? Let's see. If we pay a deeper look into it, when the chromosomes are aligned at metaphasic plate, what happens? These two chromatids, they are aligned with each other in this manner. That means they are stuck with each other all along the length. They are stuck with each other all along the length. So they need to be separated. For the separation function, what happens? Before going into that, first we need to understand why they are connected with each other. On the first hand, why these two chromatids, they are connected with each other. The connectivity or the stickiness that you are observing in metaphasic plate, that is conducted by a specified group of protein which is called cohesin. That means, suppose this is one fragment, this is another fragment. They are attached with each other. They are attached with each other by means of a protein called cohesin. Cohesin is a co-association of SMC1 and SMC3. Cohesin is the association of SMC1 and SMC3. Now, the microscopic visual of it. Suppose this is our chromatid. This is our chromatid. This is our cohesin. This may be our SMC1 and SMC3. And they are tied to each other. They are tied to each other by means of a unit. This is called Claisin. This is called Claisin. So if this entire picture, this entire phenomenon takes place by means of this, these are two chromatids, and these two chromatids, they are attached by means of cohesin, which is a combination of SMC1 and SMC3, and these two cohesins, they are attached with each other by means of a regulatory protein called claisin. This is the overall structure. Now coming to the fact why, or how rather, coming to the fact how these two chromatids gets separated. There lies two distinct kinases. There lies two distinct kinases. One is Aurora B-like kinase, and another one is Polo-like kinase. Aurora B-like kinase and Polo-like kinase. These two kinases, what it does, they activate a specified type of protein called separase. What separase does? This separase cleaves this claisin. The separase cleaves this claisin, and as a result, chromatids gets separated from each other. I'm repeating myself. There lies two distinct kinases, Aurora B-like kinase and Polo-like kinase. These two kinases, they activate a protein called separase. That separase cleaves the claisin that is attaching the chromatid units, and hence they are getting separated from each other. This is a basic phenomenon. But there lies a problem. Problem is at centromeric region. At centromeric region, this separase at centromeric region of a chromatid, this separase is blocked. This separase unit is blocked by means of another protein which is called securin. Separase is blocked by means of securin. I'm repeating myself. Initially, these two chromatids, they are attached with each other by means of cohesin. Cohesin is a combination of SMC1 and SMC3, and these two SMC units, they are attached with each other by means of a regulatory protein called claisin. Now, Aurora B-like kinase and Polo-like kinase, they are activating a protein called separase. This separase is cleaving the claisin. As a result, the chromatids are getting separated. This phenomenon in general occurs all over the chromosome except at centromere. This phenomenon occurs all over the chromosome except the centromere. Why so? Because at the centromeric region, there lies securin. What securin does, it blocks separase. And if separase is blocked, then glycine cannot be cleaved, and hence the chromosomes, rather the chromatids, cannot be separated. These two chromatids cannot be separated from each other. As a result, anaphasic movement won't appear or won't take place. So, at centromeric region, there must be certain regulatory activity going on that helps the chromatids to separate from each other. I'm repeating myself. In general, except centromeric region, all the other segments are getting separated from each other by means of polo-like kinase and aurora B-like kinase. But the problem arises with the centromeric region because there lies securin. This securin blocks the separase. As a result, glycine cannot be cleaved. So, this centromeric region, what happens over there? At centromeric region, at centromere, we have what? We have our securin. What is it doing? It is blocking the separase. As separase is blocked, hence no chromatid separation. No chromatid separation. As a result, no anaphasic movement. No anaphasic movement. But if this phenomenon takes place, then there won't be any movement of chromatids to the opposite pole, and hence the entire cell division will be ceased. In order to tackle this situation, we have CDC20. CDC20, what it does, it binds with APC by C. Now, we all have came across APC by C in my previous class. APC by C, by means of CDH1 activity, it degrades or dissociates the cyclin B. That is the regulatory aspect. So, after the activity of CDH1, the next thing APC by C does, it combines with CDC complex, CDC20 complex, and by binding with the CDC20, it ubiquitinates. What it does? It ubiquitinates our securin. It ubiquitinates our securin. And if securin is ubiquitinated, separase becomes free. And if separase is free, that will lead to glycine degradation. Glycine degradation. And if glycine gets degraded, then that will lead to anaphasic movement of chromatids. So, I'm repeating myself. At the centromeric region, there lies securin, which is blocking the separase. As separase is blocked, there will be no chromatid separation as glycine cannot be degraded, and as a result, no anaphasic movement. So, in order to rescue in this situation, there comes the activity of APC by C. APC by C, what it does in association with CDC20, it is ubiquitinating the securin. Hence, ubiquitination of securin takes place. Now, separase is free. If separase is free, it can easily degrade glycine, and if glycine gets degraded, then that will lead to our anaphasic movement. Let's see the picture over here. So this is our chromatid 1. These are the two chromatids. And this is SMC1, SMC3. They are separated, they are connected with each other by means of glycine. We have our glycine. Now, separase is blocked by securin. Whenever CDC and APC comes into action, then ubiquitination of securin takes place. If ubiquitination of securin takes place, then our separase is free. If separase is free, then glycine gets cleaved and hence the chromatids get separated. Chromatids separated. That will lead to anaphasic movement. That will lead to anaphasic movement. So this is the overall mechanism by which the chromatids get separated from each other and they can move to the opposite poles. Now this is the last topic of today's lecture. In my third part of cell cycle, we will be discussing solely upon the holistic approach of cell cycle regulation, followed by importance of checkpoints or how they are regulating the entire mechanism. That will be the third part of our cell cycle lecture. So let's see. We have, before going into it, let's deduce something. We have our cyclin and we have our CDKs. We have our respective cyclins and CDKs. And when it comes to mammalian system, CDK 4 by 6 is there. And we have our cyclin D. These are the early stage cyclins that will help the cell to enter from G0 to G1. Suppose we are considering about cyclin D and CDK 4 by 6. Now this co-association, this kind of co-association, how they are formed? How they are formed? Suppose we have a media and we are supplementing with certain growth factors or mitogen. We are applying mitogens into the medium. So mitogens, after being applied into the medium, immediately what happens? There will be activation of two sets of genes. There will be activation of two sets of genes. Pay a deep attention over here. We are entering into the genetic regulation of cell cycle. First of all, within the medium, we have mitogens or growth factors. When growth factors is applied to the medium, then two sets of genes are getting activated. One, there lies our early genes. There lies our Early genes. Within early genes, we have C-phos and C-Jun. We have our C-phos and C-Jun. Early genes or immediate genes, they are furthermore initiating the activation of late genes. They are furthermore activating the transcription of late genes. So first we have mitogen. Mitogen is activating within the cell the transcription of early set of genes like C-phos and C-Jun. Later on, this early set of genes are activating the late genes. Those late genes will furthermore help in transcription. Those late genes will furthermore help in the transcription of late G1 cyclins and CDKs. Late G1 cyclin and CDKs. Now let's see. Growth factors added to the medium. Growth factors or mitogen added to the medium to initiate proliferation at G0. So when this kind of a phenomenon occurs, immediately what will happen? Cell will activate two sets of genes. One, early response genes like C-phos and C-Jun. The C-phos and C-Jun will later on help in transcription of late genes. Late response gene, what does it encode? Late response gene, what does it encode? It encodes the mid and the late G1 cyclins. The mid and the late G1 cyclins. This is the function of our early response genes. So early response genes like C-phos and C-Jun, they will activate the mid or late G1 cyclin and CDKs, rather transcription. Now, in order to in order to function properly, in order to function properly, there lies another regulatory aspect. Suppose within our cell, when it is at G0 and trying to enter G1, suppose our cell is at G0 and it is trying to enter G1, there lies a specific protein, RB, retinoblastoma. Retinoblastoma is also stated to as a tumor suppressor gene. Now, what RB does actually? What is the function of RB? RB, it blocks E2F. RB, it blocks E2F. What is E2F? Elongation factor 2. Where it is required? At the end of G1 or at the beginning of S. For transcription, E2F is required at the end of G1 and at the beginning of S, where replication actually takes place. So E2F is rather blocked by RB in its unphosphorylated or hypophosphorylated condition. I am repeating myself. cells are supplemented with mitogens. They are activating two sets of genes. One is early gene like CFOS and CJUN. The CFOS and CJUN, they are later on activating the transcription of mid or late G1 cyclins and CDKs. As simple as that. Now this cyclins activation, the cyclin needs to be activated. Now let's go back to this section. Now this CFOS and CJUN, they are activating the late genes. Within late genes, we have mid plus late G1 cyclins. But even if they get activated or transcribed, even if they get activated or transcribed, in order to function properly, they will require E2F. They will require E2F. I'm repeating myself. Mitogen applied early genes getting activated. They are activating the late genes. Late genes means there lies mid and late G1 cyclins. As simple as that. But in order to perform these activities, or rather transcription, they will need E2F. But within the cell, that is at G0, E2F is blocked by RB. E2F is blocked by RB. So this blockage by RB needs to be removed first for the E2F to be freed and for mid and late G1 cyclins to be properly functional. I am repeating, in order to free E2F from RB, in order to free E2F from RB, there needs to be phosphorylation. There needs to be phosphorylation of RB. Hence, E2F will be freed, which will later on help in the activation or functional representation of mid and late G1 cyclins. So, RB is currently at its unphosphorylated or hypophosphorylated state. For phosphorylation of RB, what we need? For the phosphorylation of RB, what do we need? Typical cyclin, I'm stating about mammalian cells. Cyclin D plus CDK 4/6. This co-association, when we are stating a cell is entering from G0 to G1 by the activity of cyclin D and CDK 4/6, it basically means cyclin D and CDK 4/6 co-association. Cyclin D and CDK 4/6 co-association is phosphorylating the retinoblastoma protein, RB protein. And if RB protein is phosphorylated, then E2F gets freed. That means cyclin D and CDK 4/6, they are phosphorylating the RB. And if RB gets phosphorylated, then E2F is freed. E2F gets freed. And if E2F gets freed, then the mid and the late G1 cyclins can easily perform their function of transcription. And they can prepare themselves for S phase. They can prepare themselves for S phase. So this is the overall mechanism of a cell entering from G0 to G1 in response to mitogens that are activating the early genes, which are in turn activating the late genes, and by means of that, cyclin D and CDK4 by 6 gets activated, which will phosphorylate the retinoblastoma protein. As a result, E2F will be freed, which can help the transcription of late G1 cyclins and prepare the cell to enter into the S phase. So, cyclin D, cyclin D and CDK4 by 6, if present, now RB is unphosphorylated, binding with E2F. This complex is helping the RB to phosphorylate. As it is phosphorylated, E2F gets freed, and if E2F gets freed, that will furthermore activate late G1 cyclins, late G1 cyclins, so that the cells can enter the S phase. This is late G1 mechanism. When RB gets hypophosphorylated, transcription of genes like cyclin E and CDK2, and hence E2F itself gets activated so that the cells can enter from G1 to S. So today's lecture is up to this. In my next class, I will be considering a holistic approach regarding cell cycle, followed by emphasis upon checkpoints, and try to answer these two questions that I've asked during my today's lecture. And if you find this video interesting as well as informative, share with all of your batchmates. And if you want the PDF copy of this particular lecture, then feel free to comment. And always remember, stay focused, build concepts, and track progress. Me, Shanko Banerjee, founder and educator at Vital Science Institute, signing off.